It's a real honor and privilege to be a speaker and invited to speak at this opening symposium at the Hirschberg. So the title of my talk is Unraveling Immune Escape Mechanisms in Pancreatic Cancer. These are disclosures that are relevant to the presentation. Pancreatic cancer is almost always a lethal disease. It has an insidious onset, a presentation of disease that often patients have metastatic spread at the time of their clinical diagnosis, is profoundly treatment resistant, even to our most potent cytotoxic chemotherapies, such as FOLFIRINOX, which is a 4-drug chemotherapeutic regimen. While we see that FOLFIRINOX can improve the median overall survival of patients, what we find is that there's not an improvement in the long-term overall survival of these patients. And it's because of this poor prognosis and profound treatment resistance that an alarming prediction has been made that in the next 15 years, pancreatic cancer will become the second leading cause of cancer death in the United States. It's for these reasons that we need to understand the biology of this disease, and we need to come up with novel treatment strategies in order to impact the natural history of this disease. One such approach that is garnering interest and enthusiasm amongst the community and the cancer field is immunotherapy. This is based on the fact that cancer immunotherapy has the potential to not only improve the median overall survival, which is what we typically see with cytotoxic therapies, but also has the ability to improve long-term overall survival by shifting the Kaplan-Meier curve upward, as can be seen in the figure to the right. This effect, though, of immunotherapy is not a one-size-fits-all. What we're seeing is that some patients with malignancies do respond to immunotherapy, but not all patients do. And while we've seen impressive responses in melanoma, which are outlined here with various immune checkpoint inhibitors that targets the C2A4, PD-1, PD-L1 pathways, these same immunotherapeutic approaches are not demonstrating efficacy in pancreatic cancer at this time. And so this raises the point that pancreatic cancer is unique. It has devised ways in which to evade the immune system that maybe is not seen in other malignancies. And so we need to understand how is this working. And this requires us to go back to understanding what is the premise upon which immunotherapy is built. An essential tenet of cancer immunotherapy is that the immune system actively surveys for malignant transformation and can be induced to recognize and eliminate malignant cells. Well, if we look at the tumor microenvironment of pancreatic cancer, we see an environment that is quite unique. There's extensive fibrosis, poor vascularity, and some have proposed that this acts as a physical barrier to the entry of immune cells. But we certainly see that there are some leukocytes that make their way into these tumors quite readily, and they can come in close contact with malignant cells. And these are leukocytes, they're macrophages, myeloid cells, one subset of the immune system, the innate immune system. But when we look for effector T cells, which are necessary for the efficacy of current immunotherapies that are showing benefits in the clinic, we find that the majority of pancreatic cancers lack a robust infiltration of effector T cells, as we do see in some malignancies such as melanoma and lung cancer. And so what regulates the infiltration of T cells into pancreatic cancer? And how can we reverse this equation of having more T cells in and less myeloid cells, which may be causing immunosuppression in a microenvironment that is formidable to immunotherapy? One way in which we've been thinking about this is we've turned to the cancer immunity cycle that has been proposed by Dan Chen and Ira Mellman and is outlined on this slide. It states that cancer cells must first release cancer antigens that are unique and can be identified by the immune system, that these antigens are then presented by antigen-presenting cells in the lymph nodes to prime T cells and activate them. These T cells, once activated, will then traffic through the bloodstream to tumors, infiltrate those tumors, recognize cancer cells, and then kill the cancer cells. Well, if we think about the job of the cancer is to disrupt this cancer immunity cycle. And it's our job to figure out how this is happening. In terms of the immunotherapeutic strategies that are showing the most promise in the clinic to date, these are targeting two steps in this cancer immunity cycle: the priming and activation by inhibiting the C2A4, B7-1, and PD-1, PD-L1 pathways, as well as enhancing the killing of T cells within the tumor microenvironment. But I just showed you that pancreatic cancer is a disease in which we find very few effector T cells infiltrating this tumor. So maybe it's perhaps not so surprising that these therapies, which rely on the fact of T cells infiltrating, are not being efficacious to date in this malignancy. And so it makes us think about perhaps the steps in which pancreatic cancer is not initiating this cancer immunity cycle lies in the release of cancer cell antigens and in cancer antigen presentation in order to prime and activate T cells that could then be harnessed. We've been addressing this issue, as Anirban mentioned, from the standpoint of looking at the CD40 molecule. We proposed a model here in which chemotherapy could be used as a means to elicit an immunogenic form of tumor cell death, which would release antigens, and that these antigens would then be taken up by antigen-presenting cells, of which we know there are plenty of antigen-presenting cells within the tumor microenvironment of pancreatic cancer, and that these cells have CD40 on their cell surface. Now, CD40 is a member of the TNF receptor superfamily, and when it's engaged, it leads to a licensing of those antigen-presenting cells. And it was shown in the 1990s that an agonist anti-CD40 antibody could license antigen-presenting cells through this CD40 pathway. And by doing so, this enhances the processing and presentation of antigens to T cells in the context of MHC molecules, and leads to an upregulation of co-stimulatory molecules, which can then lead to the activation of T cells, which in theory would then go off and find tumor cells, kill them, and lead to a feed-forward mechanism. We investigated this hypothesis initially in a clinical trial that was reported several years ago, and I just want to make a couple points out of this trial in the next couple slides. This was a trial in patients with chemotherapy-naive advanced pancreatic carcinoma. 90% of the patients had advanced disease. There were 21 patients enrolled. We used an agonist CD40 antibody called CP870893 that had been developed and shown to be tolerable within the clinic, with the major side effect being cytokine release syndrome. We administered gemcitabine chemotherapy, which was the standard of care at the time, and 48 hours later administered the agonist CD40 antibody, with the concept being the gemcitabine would induce tumor cell death, and the CD40 agonist would then prime the antigen-presenting cells to present to T cells. This is the waterfall of the responses that were seen in this clinical trial, and we thought this was quite promising. We saw 5 partial responses, and with historical controls of gemcitabine alone, one would have expected 1 response out of 21 patients here. This is an example of 1 response. Both the primary as well as metastatic lesions were found to respond in some patients. We biopsied the 1 lesion in the bottom right-hand corner, and when we biopsied this lesion, we were expecting to find T cells within that biopsy. But what we found were no viable tumor cells, no T cells, but an abundance of macrophages. This led us to return to the laboratory, which we were running an ongoing clinical trial in mice. This was using a genetically engineered mouse model of spontaneous cancer that was originally developed by Dave Tuvison and Sunil Hingorani at the University of Pennsylvania. It incorporates 2 mutations, one an activating mutation in the KRAS oncogene and a dominant negative mutation in the p53 tumor suppressor gene. These 2 mutations are targeted to the mouse pancreas using Crelox technology, in which Cre recombinase under the control of the pancreas-specific PDX1 promoter unlocks these 2 mutations. These mice spontaneously develop pancreatic cancer, and they develop many of the same hallmarks that we see in human pancreatic cancer. We see a desmoplastic reaction. We see deposition of matrix proteins, COL1, type 1 collagen, amidst the EpCAM-positive malignant cells. We see an abundance of macrophages, F480 positive cells that are recruited, and we see an exclusion of effector T cells, CD3 positive cells. We repeated the clinical trial in the mice Here the mice were imaged using ultrasound. We treated the mice with the same treatment strategy, gemcitabine followed by an agonist CD40 antibody, which is the FGK45. The waterfall plot shows that we saw very similar responses as we saw in patients, about a 20-30% response rates. This was reproduced with an agonist CD40 antibody alone. We asked whether or not this was T-cell dependent, and unfortunately we found that our hypothesis was not correct. But neither CD4 nor CD8 or the combination were required to induce this efficacy that was seen with an agonist CD40 antibody alone. In fact, when we looked for T cells within the tumor microenvironment, we didn't find T cells infiltrating either. In fact, they remained confined to the tumor draining and peripancreatic lymph nodes and never made their way into the tumor. And so this led us to ask the question of how is pancreatic cancer excluding T cells? This was a treatment approach that had been shown through multiple implantable models, and we showed it as well. It could work to drive T-cell dependent immunity, but it wasn't working in this model. Is this because of the poor antigenicity of the model? There are only two mutations, one in KRAS and one P53. And certainly, human pancreatic cancer also lacks a significant number of somatic mutations, and hence there may not be very many neoantigens that are presented in this disease as one sees in melanoma and lung cancer. Pancreatic cancer may also be poorly immunogenic. Little MAC expression means less presentation of antigens, the ability to secrete cytokines such as TGF-beta that may be suppressive to the T cells. And certainly there's an immune suppressive microenvironment of cells that are present, and then there's this potential physical barrier, the fibrosis. We initially addressed this question by looking at this barrier. And to do so, what we did was to take the KPC mice, explant a tumor from one mouse, and take chunks of that tumor and place it under the skin of a second mouse that had a primary tumor in the pancreas. This mouse now has two tumors. We now treated this mouse with a control treatment or our vaccine strategy, gemcitabine followed by an agonist CD40 antibody. Now the advantage here is that compared to cell lines, the explant tumor maintains that microenvironment that one sees within the primary pancreatic tumor and allows us to now address whether or not we could get T cells through that fibrotic reaction. And what we found was with this treatment, 14 days after treatment, we found 50% of the animals, and figure C there, had responded with complete regression of their tumors on the explants. And if we looked at the explant histologically, we found that T cells were infiltrating. It didn't appear as though this was the microenvironment was a barrier. In fact, now that T cells were infiltrating and being induced against this explant tumor, we could find T cells infiltrating the primary pancreatic tumor as well, but not all T cell subsets infiltrated, suggesting that there's a local regional mechanism of that's causing preventing CD8 T cells from infiltrating into the tumor. And so we hypothesized that perhaps this is related to that inflammatory reaction that we see within the tumor microenvironment. Certainly macrophages have been shown to be involved in many of the hallmarks of cancer that have been proposed by Doug Hanahan and Bob Weinberg. And avoiding immune destruction is one of these mechanisms that macrophages have been shown to be involved in. And so we asked the question of whether or not macrophages may be regulating the infiltration of T cells into tumors. And I already showed you that gemcitabine followed by an agonist CD40 antibody in this KPC model is unable to induce T cell infiltration. And so we depleted macrophages using an approach that involves liposomes that are encapsulated with cladrionate. Liposomes are eaten by the macrophages, and cladrionate is toxic to the macrophages. And using this approach, we can find depletion of macrophages within the peripheral blood, the spleen, the liver, as well as peripancreatic, peritumoral lymph nodes. And when we combined now this cladrionate encapsulated liposome approach with chemotherapy and an agonist CD40 antibody, we now found T cells infiltrating into these tumors. In fact, if we looked at the subsets of cells that were infiltrating, we saw that both CD4 and CD8 T cells infiltrated. And in fact, even just delivering the macrophage depleting regimen elicited T cells to infiltrate into the tumor microenvironment, but not both subsets. It was only the CD4 T cell subset. This implies that there's an active mechanism that's being regulated by these macrophages in terms of the infiltration of T cells into the tumor microenvironment. And so we asked whether or not this had any treatment efficacy, and to do so we ran a clinical trial in the mice. This is a waterfall plot looking 14 days after treatment. In control-treated animals and animals depleted of macrophages, we continued to see progression of disease. The agonist CD40 antibody led to regressions in about 30 to 40% of animals. This was blocked by depleting macrophages, which we had previously shown. The macrophages in this setting we had found were important for, they acquired an anti-stromal as well as a tumoricidal capacity. If we combined gemcitabine and agonist CD40 antibody, I showed that we were able to see regressions. If we depleted macrophages in this setting, though, we still saw tumor regressions. But now we have T cells infiltrating these tumors, and so we asked whether or not T cells are required, and depleting T cells with an anti-CD8 antibody led to an abrogation of this effect, demonstrating that T cells can infiltrate the microenvironment, this dense desmoplastic reaction, and that they can mediate anti-tumor efficacy, even in a model that is initiated with only 2 mutations. So we wanted to know which macrophages are being targeted. To do this, we used liposomes that were fluorescently labeled and administered them to the mice. And one of the important things that we noticed was that the liposomes did not infiltrate the tumors and label macrophages within the tumors. They labeled macrophages in the spleen, the peritumoral lymph nodes, the blood, as well as the liver. And when we looked at the peritumoral lymph nodes, remembering that we thought that this was a local regional effect, that's the mechanism of resistance, we found that the liposomes were co-localizing with macrophages in lymph nodes in a particular location within the subcapsular sinus. When we phenotyped these cells, we found that they lacked GR1 antibodies, Ly6C and Ly6G, which are commonly used to describe myeloid-derived suppressor cells. And so these macrophages are a unique subset. They're not tumor-associated macrophages, they're not myeloid-derived suppressor cells, and they're cells that are lying outside of the tumor microenvironment. In fact, these macrophages are anatomically located in a spot that scientists who study autoimmune diseases know a lot about. They're located in a region where they capture antigens and are important for inducing tolerance. And so we're now investigating the mechanism by which these macrophages may be regulating priming of antigens within the peritumoral lymph nodes. This raises the point that pancreatic cancer has devised ways in which to regulate potentially the release of cancer cell antigens, their antigen presentation, and that much of the regulation may be taking place at the level of priming and activation. This is consistent with data from the Johns Hopkins group looking at vaccine strategies in which they induce an immune response, and they can now see T cells entering into the tumors. But we have another way now that we can actually address the mechanisms of the other part of this cancer immunity cycle, and that is adaptive transfer of T cells, the infusion of engineered T cells. This approach we've been using at the University of Pennsylvania involves the development of chimeric antigen receptors. These are a fusion of an extracellular domain and an intracellular domain. The extracellular domain is comprised often of a single-chain variable fragment from a tumor surface antigen antibody, which is fused to the signaling domains of the T cell receptor and co-stimulatory signaling domains such as 4-1BB or CD28. This CAR is then inserted into T cells, and when it engages its target, which is a surface antigen on the surface of a tumor cell, it will now send a signal to that T cell to activate it and engage it to kill the target cell. This has been most extensively evaluated to date in hematological malignancies, where impressive responses have been seen in acute lymphoblastic leukemia, with greater than 90% of patients showing complete responses in heavily pretreated situations. And the targets have largely been CD19, although others are being explored. What one will notice is that all of these antigens are self-antigens, with the exception of EGFR variant 3 under the solid tumor column. And this is a challenge as we think about translating CAR therapy to solid malignancies, because one has to be concerned of the potential for on-target but off-tumor toxicities. We've focused our efforts to date on mesothelin, which is expressed on the lining of the pleura, the pericardium, and the peritoneum. For these reasons, we've developed two expression platforms. One is an RNA approach, which leads to the transient expression of CAR within a T cell. And the second is using a lentiviral approach, which leads to a permanent DNA integration of the CAR into the T cell. We've used a single-chain variable fragment derived from the mouse anti-human SS1 antibody that was developed from Ira Pastrin. This single-chain variable fragment is fused to the signaling domains of the 41BB and CD3-seated chains. With this approach, we've evaluated first in vitro the specificity of this strategy, and one can see that from a CTL assay that the CAR T cells engineered to recognize mesothelin show specificity against the pancreatic cancer cell line BXPC3, which expresses mesothelin, but do not lyse K562 cells that express CD19 and lack mesothelin. We've evaluated preclinically the potential in vivo activity of RNA versus lentiviral CARs in NSG immunocompromised mice. And the tumor model is a human mesothelioma cell line. And one can see that the RNA CARs do have efficacy, but the lentiviral CARs are certainly superior given their sustained expression of the CAR. We've translated this now into the clinic in a phase 1 study. And our approach is to apheresis patients. Lymphocytes are separated. They are then engineered and expanded, and then quality controlled and finally reinfused back into the patient. This manufacturing process takes about 3 to 4 weeks to complete. Patients are then infused with CAR T cells, RNA CAR T cells intravenously 3 times per week for 3 weeks for a total of 9 infusions. With this approach, we've seen in vivo presence of the RNA CAR T cells immediately after infusion, but their persistence is transient as one would have expected. When we've looked to see if there's safety, we've actually found no on-target off-tumor toxicities. They've been well tolerated in all 6 of the patients that have been treated thus far. In one of the 6 patients, we've seen evidence of potential anti-tumor efficacy. This is a patient that at baseline had disease within the liver as well as the primary pancreas that can be seen here on FDG PET-CT imaging. 1 month after treatment, one can see that there's a complete loss of FDG uptake within the liver. There's a decrease in the signal within the pancreas, although the lesion has actually swelled here. If one looks at the CT images, at baseline, the red arrows show lesions. After 1 month, one can see that these lesions have also swelled, but have lost the FDG avidity. Based on the safety and potential efficacy, we've translated this now to using lentiviral CAR T cells. We've also seen safety with this approach thus far in the patients that we've treated. We've also now seen that these cells expand within the peripheral blood, but their persistence is transient. And this brings us back to the potential that there could be an immunogenicity issue. This is a mouse antibody that's used. Alternatively, this may point to features of the macroenvironment of the patients or alternative intrinsic features of the T cells that are being infused, which come from the patient and have been exposed to prior to chemotherapies, as all of these patients are heavily pretreated. Overall, we are focusing on 5 elements, and we're using this approach as a tool to now unravel mechanisms of immune escape that occur not only at the level of trafficking, but also at the level of the tumor microenvironment. We've seen safety and feasibility thus far. We've seen the persistence expansion is transient. We have seen trafficking of T cells, CAR T cells into tumors, which we can detect by QRT-PCR and PCR for the RNA and lentiviral trials, respectively. We've seen some evidence of clinical activity, which I've shared with you, but ultimately we believe that the tumor microenvironment is another mechanism of immune escape, and that there will be elements of adaptive immune resistance and immune suppression that exist there that we'll need to overcome, such that combination therapies are likely to be needed. Together, what I hopefully have shown you is that pancreatic cancer, we believe, is targeting multiple aspects of this cancer immunity cycle, that there's mechanisms of immune evasion that occur at the level of antigen release, antigen presentation, priming, and activation. There's trafficking and infiltration mechanisms that are at play. And understanding each one of these mechanisms, we hope, will provide rationale for how to combine therapies that are already existing in the clinic, but doing so in a rational way in order to improve the outcomes for patients. So I'll end here and just acknowledge the many people who have been a part of this work, members of my laboratory, as well as my clinical colleagues, Peter O'Dwyer, who was involved, and Bob Vanderheid, was involved with the CD40 trials, Andrew Haas and Yanis Tanya and Mark O'Hara, who are involved with the CAR-T-cell work, the radiologist who's done the PET-CT imaging, Drew Teresian, our cell vaccine production facility led by Bruce Levine, and our Corridor Lab Studies group led by Josef Mellenhorst, and also Carl June and his group who have pioneered this work at the University of Pennsylvania, and our funding that has helped to support this work. Thank you for your attention.